Good morning, Claude.
Hey, good morning. How are you?
Yeah. How are you today?
I'm good. It's still a bit early for me, but it's okay. Let me go
We'll
to a
start
place.
shortly.
Okay,
We're just waiting for a few more people to join. Thank you for joining us.
my pleasure. Do you want to tell me how do you want to proceed after the talk? If you want me to hang around for a while, or
I was thinking we could do that after the talk.
okay. So just stay with you and on this link after the talk.
Yeah.
Okay. Maybe a while. Let me get a charger. Make sure I don't lose you for like hour. So who here? Can you hear me, Tanya?
Yeah, I do.
So tell me, who are you? Who are you exactly?
So I'm actually the Deputy Chief Operating Officer here at Project Encephalon. I'm a master's student. I'm doing my thesis work on the gut-brain axis at NISER Bhubaneswar.
Working on mice?
Yeah. I work on the mice models,
Okay.
C57 and BALC.
Okay. So let me see if I can share my screen. And you are located in Calcutta?
No, I'm actually in Bhubaneswar. Calcutta is not far. I used to live there a few years back. Now I'm in Bhubaneswar.
Yeah, Bengali. You speak Bengali, or you? Yeah.
I know a little bit of Bengali, but I'm actually Odia.
Okay. So let me share my screen. Where's my screen? And my audience is what exactly? People like you, or?
Excuse me?
Who is my audience?
Oh, audience. Okay, so we all are Project Encephalon members. Some of them are from high school, but most of them are undergrads and postgraduate students.
Okay. So they will understand if I go to talk. So I should keep it simple. Say some high school kids. Yeah.
No, you can go into the technicalities. I'm sure if somebody has any doubts, they'll ask.
Okay.
Okay.
For example, do you know what people know about single-cell RNA sequencing?
I think some would know. Okay. So should we start?
Whenever you want, with me, yeah.
Okay. So good morning, evening, and afternoon, as and when I find you guys. So welcome to the final day of Cajal week. So we are here celebrating the 169th birthday of Ramoni Cajal and celebrating the creative pursuit of neuroscience as it is. So today we have with us Dr. Claude Desplan. It gives me immense pleasure to welcome him. He has this really great profile that I'm sure you all already know. So he is currently a Silver Professor at NYU in the Department of Biology and Neuroscience. He works on unraveling the development of color vision in the Drosophila model. He was born in Algeria and trained in France. He received his BSc at INSERM in Paris and then joined Pato Farrell in UCSF as a postdoc. And now he is currently in NYU and he's here to give a talk on the generation of neural diversity. So over to you, Claude.
Thank you. Thank you, Tanya. And let me share my screen. And like that. Okay, so good afternoon or good morning or whatever. And it's very early in the morning for me because it's 7:30. I'm in New York. So I'm a professor of biology in NYU, New York University. And I teach quite a bit actually, and mostly teaching undergraduate. And I also have a lab at NYU. Actually, I also have a lab closer to you, not that close, but in Abu Dhabi, because New York University has a branch in Abu Dhabi where we have a good number of very international students, about 120 nationalities, who study there toward different degrees. So my lab, both in New York and in Abu Dhabi, are dedicated to understanding how you can create the huge diversity of neurons that you can see in your brain. So the next picture could be a picture from Harmony Cajal, but because basically this image of different types of neurons, which have been stained using this amazing technique that Cajal did not invent, but used extensively, called the Golgi technique. And it's a kind of a miracle technique whereby you treat the neurons, the brain, with some silver solutions, and some of the neurons take up these solutions and they stain the whole ensemble of the neurons. And what you can see here is that the neuron can acquire very, very different shapes and forms. And so just a small example of many of the neurons that you can figure out. The very long neurons, not very one branch, or extremely long branch, extremely branched neurons here. Okay, and so this is huge diversity of morphology of neuron and of course of function. But in fact, we don't study the neurons in vertebrates because it's too complicated. Instead, we use the fruit fly Drosophila eyes to be able to understand how this happens. Not so much the eye, but the eye and all the system of processing centers that are behind the eye that process visual information. And the advantage of this system is that it has that great advantage of being much, much simpler than what we know from human. As you know, in human, we have about 80 billion neurons, a very large number of neurons, and each one of them make about 10,000 connections to each other, which means the number of connections is huge, on the order of 10 to the 14 or 15. And we have absolutely no idea how many cell types we have in a human brain, no ideas. And the more you dig, the more you find neuron types. And so we don't know because we don't have any type of systematic elaboration of how many neurons we can figure out. In sharp contrast to that, the Drosophila optic lobe has many, many fewer neurons, about 1 million times fewer neurons, because it only contains 60,000 neurons. And in fact, this represents the majority of the fly brain. So fly brain is maybe 80 or 100,000 neurons, which means most of the neurons in a fly brain are dedicated to vision. Those animals are very visual. They can fly high speed, do acrobatic flying, and therefore they need a very, very good and fast eyes. And therefore, most of their energy is used to function for the functioning of this optic lobe, the place that process vision. Again, starting with Harmony Cajal about 100 years ago or more, we now have a very beautiful description of most of the neurons that contain that are present in this optic lobe. So Cajal didn't work on the fruit flies brain, but he worked on some bigger flies brain. And he basically looked at the neuron of almost every species he could put their hands on. And of course, he didn't choose the smallest one. He chose the biggest fruit flies, house flies that you find at home. But the organization is very, very similar, except a bit larger in the context of those bigger flies. Anyway, so we know that we have about 200 neuron types. And this is just based on morphology. And I would argue that it's not many more than that for a reason I would just to explain to you, okay? On top of that, the HHMI, which is a Harvard Medical Institute in Janelia in the US, has managed to establish the connectome of all those neurons. What I mean by that is that they did electron microscopy reconstruction of this brain structure, and they've managed to establish which neuron connects to which neurons, and to do that in a very extensive manner, which means we know the neuron types, again, starting with Cajal, but we also know how they're connected to one another. And they could do that, as you will see why, because the optic lobe is a very a structure which can be made of repetitive structures, and if they understand one part of the optic lobe, they can understand the whole system of the optic lobe. But in spite of this very small brain, 60,000 neurons, again, the flies can do flies very well and very high speed and can really do very well in terms of the structure. So I'm going to show you on the right is a picture of not Cajal, but actually somebody who really took advantage of the work from Cajal, and but 80 years later did actually a huge amount of work to define all these cell types. This guy Fischbach, he was in Germany, and what he did, he basically identified by morphology most of the neuron. And then what we did, we took all of these pictures and assembled them into something that looks like a network. Okay, so it's just for illustration purpose, and you can just see here that you have neurons of different types, and this is describing all the neurons that form the optic lobe. So even if it's not as complicated as the human brain, you can still that is very complicated when you know all those neurons, and there are only 200 of them are piling up on each other and connecting to each other. But again, thanks to the electron microscopy reconstruction, we know how basically each one of those structures here is organized and how the neurons are connected to one another. So let me give you a description of the fruit fly visual system to tell you exactly why it's such an advantageous system. Okay, so it's a picture, it's a real picture from fruit fly eyes, and you can see it's organized in different layers. The first layer is in fact the eye itself, and the eye is made, of course, of photoreceptors, and the fruit fly eye doesn't have a very high resolution. As you might know, the fruit fly eye is made of it's the compound eyes, which means it's made of these different facets. And each facet has its own lens, and each facet basically looks at one point in space. In fact, there is about 800 facets in the context of the fruit fly eyes. And all insects have this limitation that they have to make facets for each pixel that they detect. And the best resolution animals, maybe the bigger flies or butterflies or have a maximum resolution about 10,000 ommatidia and 10,000 pixels. Yet, with only 800 pixels, the fly eye can do very well. What we have done here, so those investigators have done here, is to label the photoreceptor in green. And you can see that if you were to count, there will be about 800 groups of photoreceptors, each one detecting one point in space. Those photoreceptors are also neurons, but that means that they also send projection to the brain. And you can see those projections here in green, they go down, and they go down to the next layer of the visual system, behind the eye. It's called the lamina. And the lamina is going to have 800 cartridges. They correspond to the 800 ommatidia, the 800 unit eyes, 800 facets that you see in the eye. This structure is actually dedicated to the detection of motion. You know, when you fly at high speed, you need to be very careful to know exactly where the world is and not bang into any object, and therefore you need very good detection of motion. And so this is a fairly simple structure whose purpose is, before all, to detect motion. And so there is and you can see here that the green photoreceptor project into this lamina part, and they're going to connect to lamina neurons, and those lamina neurons are going to go from the lamina to the next layer of the optic lobe, which is called the medulla. Okay? And so the medulla receive input from the lamina neuron, which I don't show here, but what I see in green actually some other photoreceptors, because not all photoreceptors coming from the eye are stopping in the lamina. Those involved in motion vision, which are like the road on our retina, the black and white photoreceptor of our retina project to the lamina, but the equivalent of the cones, which means the color photoreceptor of the fruit fly eyes, they go through the lamina and they stop in the medulla instead. And you can see here in green again, 800 columns corresponding to the 800 pixels detecting by the eye here. And then they need to get projection to higher processing center. I will not tell you too much more about it. We do a lot of work on that, but it's not the purpose of my talk today. Okay? So the beauty of the system is that, as I was telling you, that each pixel is going to project to the lamina, to the medulla, and to the high processing center. And the pixel which is here is going to project to the pixel that is here. And what you're going to have 800 circuits running in parallel to each other from visualizing the whole world. And the beauty of the system is because if you can understand how one of those circuits here, that you have the one, for example, described here, if you understand how the circuit is built and how it functions, you understand the whole thing, because you just need to extrapolate to all the other pixels, also the circuit running in parallel to this one. And it's why actually the Howard Hughes Medical Institute was able to reconstruct this structure here, because they just took a few cartridge, a few columns beyond that, and understood how they connected, and they could extrapolate to the whole thing. So we start with a huge knowledge of all the neuron types that exist in this structure and all the connection amongst those different neurons. But this is a purely morphological description, a bit like what Cajal was managing to do. It doesn't tell you exactly how the system functions, and more important for us who are developmental biologists, how the system is being built. And I believe that if you understand how the system is built, you understand a great deal about how it's going to function. So let's see what type of neuron exists in these structures. In the eye, there are about 20 cell types, mostly photoreceptor and support cell photoreceptors. And again, I told you there is rod-like photoreceptors that project to the lamina, and cone-like photoreceptors that project to the medulla. The lamina is a very simple structure. It only contains 5 cell types, 5 lamina neurons that are involved. All of them are involved in motion vision. And they basically process how an object moves from one place to another. And it's a very simple structure. But the complexity occurs in the next step of the pathway, which is the medulla, which is composed of at least 100 cell types. Because what the medulla does, it does every processing. It processes the color vision here because it receives the input from the color photoreceptor cells. It also receives the motion vision from the lamina. And then it's going to mix all of this, process all of this, and it has a lot of neurons that are being there. What I mean by that each one of the columns that you see here, each one of the columns is surrounded by 100 cell types that are going to process the information. So very extensive processing. Then you're going to project to the lobula, and the lobula is actually where you're getting the output of the system. Okay? So question we want to see is first, can we describe all those neurons in now more molecular terms and understand a bit beyond what Cajal and Fischbach were going to do, beyond the morphology, but to try to understand how the gene expression can control all of these phenomena. So first we need to describe those neurons precisely. And again, switching from Golgi staining a la Cajal to GFP stainings, we could use modern technology of clonal analysis, but you can label one single neurons, for example. And so I forgot to tell you, but if we focus our attention on the medulla, because it's the most complex structure, you can see that the medulla is formed of blue region and a red region. And the blue region is simply the cell bodies of those 100 cell types. And you have about 40,000 neurons, which are present here, 40,000. And again, as you remember, they are projecting to 800 cartridge. So if you're good in math, you say 40,000 divided by 800, which means it's about 50 to 100 neurons that surround each one of the cartridge. So now what you can do, again, to analyze it, you can do what we call sparse labeling, which means you can label a single neuron. And again, we have beautiful genetic means of doing that in fruit fly. You can take one neuron, for example, this cell body, and see what it's doing. It's a bit like doing what Cajal was doing, except here we can control a bit better what we're doing. So I'm going to show you a neuron that has a cell body here in the blue region. It's going to project to these layers of the medulla, then to that layer of the medulla, then to the red region. And the red region is basically all what we call the neuropil, which means all those connections among the branch of the neuron. There is no cell body, there is no nucleus. They just process axon and dendrite. They talk to one another, either talking to the photoreceptor in that region here, or just talking to each other here in this region. So I'm going to show you one of those neurons here to label, and you can see that this neuron has a cell body again in the blue region here, and you're going to project to the red, which is the photoreceptor. So basically what is in green here. Sorry, I changed the color. And it makes arborization, which are dendritic arborization, connect to the photoreceptor. Then it goes a bit deeper to another layer of photoreceptor in red. And then it makes arborization into this region where there is processing, which is most likely it makes connection with many other neurons in this region. And then we need to project through this single process here to the higher processing center. I should tell you that flies are a bit different from vertebrate neurons in the sense that often they are monopolar, which means there is only one branch coming from the cell body. And from this branch, you have dendritic hybridization, and then the main branch becomes the axon itself. So that means that, if you know a bit of neurobiology, you know that processing of information within one neuron occur often in the cell body. Here, the processing occurs at this level here, so it's a bit different. But overall, the principle remains the same. So as you can see, most likely these neurons correspond to one of those, and it corresponds to one of the columns that you have in the medulla. We call this unique columnar neuron because we believe there is one such neuron for each one of the 800 columns, which means most likely you're going to have a structure like that, where each column is innervated by one of those neurons, and you expect to get about 800 of this type of neuron, okay? Unique columnar neurons. But of course, you cannot process the information just line by line because you need to be able to have a broader picture of the world. So you have other neurons which are collecting information from a set of neurons which are photoreceptors which are next to each other, a set of columns. So this is called multi-columnar neurons because these neurons collect information for about 10 cartridges here and here, again in red as a photoreceptor, processing here, and then you're going to send it to higher processing center. So now, if you want this neuron to cover the whole receptor field, that means that you should see something like that, where they touch each other and process information from a bunch of cartridges, which means now you're going to have fewer of these neurons than in the previous case, because they have cover larger receptor field. So you might imagine, for example, they are 75 of those neurons instead of 800 of these neurons. Okay? So we did that over and over again, and again, HHMI, Howard Hughes Medical Institute in Janelia also did that, and the field did that, and we could have a compilation of all those neurons. And let me tell you how they are distinguished. So first you have what we call projection neurons, and projection neurons are basically neurons that collect information from the photoreceptor, for example, and then send it to a higher processing center. They project outside of the medulla, for instance. And you can see the projection neurons that have long like that. And as I was telling you, you have also local interneurons, which are neurons that don't get out of the medulla or don't get out of the lobula and process information locally. They are short, they remain there, they don't project outside. But for our interest also, what is more important is to distinguish between the unique columnar neurons, with those which are very narrow, they only connect to one cartridge, and you expect to get 800 of these neurons, okay? As compared to the multi-columnar neurons, which are fewer in number because they cover larger receptor field. But there is about 25 cell types which process information in different way in each unique columnar cell type. And there's many more types of multi-columnar neurons, about 70 of those cell types, and together 70 plus 25 makes about 100 neurons, okay? That means you have 100 cell types that process information in a multi-columnar manner. So now what you have to do is to figure out how can you generate this type of diversity, 25 unique columnar neurons, we need each 800 of each, and 70 types of multi-columnar neurons. And those neurons, in fact, cover a different region. And some of them are 75 of each, but sometimes are about 5 of each, some are 600 of each. So quite a variety depending on the size of the receptor field, how much they cover of the receptor field. So how do you do that? So again, about 5-6 years ago, when we started doing this work, we had actually tools, again, available in fruit flies, which are modern tools of molecular genetics, whereby somehow we could manage to find a gene that was expressed specifically in only one cell type. For example, a gene only expressed in this cell type and not expressed in any of those cell types. And we can make a reporter for this gene, and we could actually label very specifically in the brain only one cell type. Okay, so well, you have an example here whereby you can see one of the neurons. Okay, it has these neurons in the cell body in the blue region. Okay, and then it projects to the photoreceptor layer here, and you can see the projection neurons, and is in the medulla, and it's going to project to the lobula here. Okay, so you see project to the lobula. And therefore, now this brain only contains those GFP neurons. So what you can do is dissociate this brain and then use a fax machine to be able to sort the neurons that are GFP positive. In such a way, you can collect very painfully quite a large number of neurons which are all the same. Okay, this type of neurons you have here. And then once you have all these cell types, what you can do is actually sequence them, send them to make a cDNA, and then sequence the transcriptome of those neurons. So you can do that for 1 neuron, for 5 neurons, for 10 neurons, and altogether the field has done it for about 50 neurons. But as you can imagine, it's a very painstaking process because each time you have to find the right line expressed in one neuron only, and then you have to fax them, and then you have to sequence them. So this was quite difficult, but the field managed to do that. And again, between our work and the work from the Harvard Medical Institute, we could sequence a good number of those projection neurons, a good number of those local interneurons, and of the multicolumnar neuron as well. Okay, but this was only a small, maybe 50 out of 200 neurons that you have in the whole optic lobe. Okay, this is not only the medulla; it's the whole optic lobe. And so, this is about 4 years ago that a new technology appeared on the market, which was called single-cell sequencing. And if you are going to be neurobiologist, you cannot escape the single-cell sequencing because it's an amazingly powerful technique that allows you to really understand how the neurons are being made. So what is single-cell sequencing? As I told you in a previous slide, I told you that you can sequence each one of the neurons one by one by purifying them and making, maybe collecting maybe 5,000 of each of the neurons and sequencing them. But since then, the single-cell technique consists of being able to define the transcriptome, the RNA content of a single cell. So what you do in this case, you just dissociate your brain, and what you're going to do is to take each cell one by one and put that using microfluidics into one droplet of oil. And then what you can do is that do some chemistry in the single droplet and sequence the RNA which is present in this single cell. It's an amazing technique. And what you can see is that often detect up to 5,000 to 10,000 transcripts corresponding to a few thousand genes that are present expressed in this in this cell, single cell. So now what you can do is have a very precise vision of all of the neurons that present, because if you have a single cell, you can sequence it and you can do it. So of course, when you do that, if you have to sequence from each one of the cell one by one, it's very difficult to have a thorough description of all the RNA expressed in this cell. And therefore, what you have to do is to sequence many of the same cell types and then try to group together those cells that look the same. And there's a lot of very complicated bioinformatics to do that, in particular a program called SORAT, which is developed by our colleague at NYU called Rahul Satija. And what SORAT does is just process bioinformatically the information coming from each one of the cells that you did and group cells that look the same. And although you cannot have a full description of the transcriptome from a single cell, if you put together all the cells that look the same, you can have very precise description of all the genes expressed in one cell type. So you do what we call clustering, which consists of putting together onto a bioinformatic map all the neurons that remain that look the same. Okay, and you have a picture here of what this resembles to. So what you can see here is that each dot here is a single cell, which means in fact you have a very large one. You have about 100,000 cells that have been each one sequenced individually. So each dot is one cell, and the transcriptome of one cell. And what you have done here is group them by neurons that resemble, whose transcriptome resemble each other. Okay? And so this is, of course, very large amount of work and 110,000 cells. And it could look quite expensive to do that because you have to sequence each cell one by one. But there are a lot of tricks that actually make this, I would not say cheap, but affordable. And you can really sequence a large number of cells. And now people, for example, at the Allen Institute in Seattle, have sequenced millions of neurons from the human brain of the mouse brain. So what you can do is group those cells based on their transcriptome. And the one huge disadvantage to the system is that you do know the transcriptome of one cell type, but you don't know what this cell type is because you have lost completely the information about the morphology in doing the process. You dissociated the cells, you put them into a droplet, and of course, at then you no longer know what the shape of these cells was. Which means you have the transcriptome of neurons, but often you don't know what those neurons are. But you can still recognize a neuron from a glia, for example, based on its transcriptome. You have genes which are very specific to neurons, genes which are very specific to glia. In this case, I mean, now we have increased this number. We have about 200 neurons indeed, and about 13 to 14 types of glia. But you remember on the previous picture, we and others have identified the transcriptome of a large number of neurons based on, bulk sequencing of the FACS peripheral neuron, which means somehow we can put a name to some of those clusters. And of course, you can see that the clusters are different in size. So you have large clusters like this one, like this one, like this one, this one, and you have tiny clusters like this one and this one. And of course, if I remember what I told you there is two types of neurons in this brain. You have the neurons that are what we call unique columnar neurons, which means the neurons that are present in every cluster, and there is 800 of them. And you have the multi-columnar neurons, which are much less abundant. And of course, you can see that the large clusters, which are all about the same size, correspond to those unique columnar neurons, those neurons that are present in large abundance, and therefore you recover them more often, and therefore you have more cells which are of the orange type, or the blue type, or the green type here, okay? And indeed, we could put a name on those genes because they match what we had purified from the bulk sequencing. And you can put a name on that, and those neurons indeed correspond to unique columnar neurons that you find in your image. And then you can try to do the same thing for the multi-columnar neurons, the smaller cluster. It's a bit more difficult, but you can also figure this out. And so what you have been doing over the last year since we did this work, is that actually try to look at the transcriptome of cluster which don't have a name, and find genes which are very specifically expressed in this cluster, in none of the other clusters, and try to find a way to see in which neurons, morphological neurons, you can see this gene is expressed. And again, there are tricks for doing that, but we managed to do fairly slowly to identify all of those cell types one by one. And now we have about half of the neurons in this picture are known in terms of their morphology. And of course, you can realize there is a hugely powerful system here, because now we know how each one of the cells is able to achieve its function through its transcription, because we can match the transcriptome and the morphology, as well as a connection of this neuron to other neurons. So for example, we could figure out what are the transcription factors that control the shape of the neuron, or what are the transcription factors that control the connection of the neuron, what are the cell adhesion molecules that allow two neurons to connect to one another, and so on. However, there is a limitation here that, and I'm going to illustrate it by showing you this big group of blue cells here, okay? Usually in these techniques, when you find a blob of cells, a good group of cells in the middle, they are what we call crabs. That means, there are neurons you cannot identify precisely. Basically, there is a group of RNA that leaked out in the medium, and you dismiss them. However, in this case, we cannot dismiss those neurons because, in fact, they match very well a group of 8 neuron cell types. They're called the T4 neuron and the T5 neuron, and each one of those neurons belongs to 4 subset types. That means there is T4A, B, C, and D, and T5A, B, C, and D. And those are indeed very important neurons because those are neurons that can detect motion. The first neuron they can detect motion, okay? And they can detect motion. The A is front to back. When the fly flies at high speed, the world around it moves from front to back. When it turns right, some neuron will see front to back, the other eye will see back to front, but the fly can also go up and down. So A sees front to back, B sees back to front, C sees up, and D sees back. And what TA does, as T4 does as compared to T5, T4 can see motion of a bright object on a bright background, while T5 sees the motion of a dark object on a black background. Okay, so they go, and you do the same thing in your own eye. You can detect either dark object or white or light object. And again, you can see them in different directions. We don't need too much to see in the up and down because we don't fly, but the fly need to do that in the four-dimensional space, four-dimensional space. So what was surprising here is that although we knew there was 8 neurons, they all were exactly in the same blob. That means they all look the same in terms of the transcriptome. And we thought about it and we realized that maybe this is not so wrong. Maybe it makes some sense. Because as you can understand, those 4 neurons, what they do is they compare 2 points in space, for example, 1 point here and 1 point here, and they see whether the world move from one to the other. And so what they do is they do the same thing. They do compare 2 points in space. It just depends how those 2 points in space are connected. They could be that way, or that way, or that way, or that way. It can be front to back to front, up and down. And of course, in the adult, they function the same way, but they are wired differently. So we thought maybe the problem here is that we look in the adult. In the adult, everything is connected, and therefore everything in this case can function the same way. So all those 8 T4 neurons have the same transcriptome because they function the same way, but they're connected differently. So we thought, indeed, if you want to get the full extent of diversity of neurons, you need not only to figure out how they do that in the adult, you also need to do that to understand what I do in the context of development. Okay. So we say, let's try to do the very same thing, except let's do it during the steps of development when those neurons are making connection. So those neurons make connection. So this is the timeline of development. The fly starts the larvae, then becomes a pupa, and then it's going to eclose into an adult. And we know the transcriptome of the adult neurons here. But the projection to the different layers of the brain and the formation of synapse occur much earlier at 50% of development here during the pupal life. And so we say, if we could maybe identify the transcriptome of those neurons at this stage of development, we might be doing a great deal. However, there is a problem. You can do the sequencing, you can identify, you can make those clusters in here, but the problem here is that those neurons again are going to have very different transcriptome than those of the adult, because they are just developing, they're just progressing, and therefore it will be very hard to know which neuron you are sequencing here because they don't very much resemble the neurons in the adult. But we thought maybe we can do something that we can take the neurons in the adult and then look a bit slightly earlier in development, for example, at 70% just before they close. And of course, this should be resembling the adult neuron because they're going to become adult neurons. And so we can use techniques that allows us to compare these neurons, this cluster at 70% of development with the adult, and then manage to identify what's going on, what the name of the neurons which are present at 70. And then what you can do is go back in time, back and back and back, all the way to the earliest time of development. So you could not match the neuron at 15% of development with the adult. But if you do that step by step, you can indeed do that. Okay. So we did that and it worked actually beautifully well. So what you have here is in a picture, the same picture I showed you before, okay, with the huge blue cluster of neurons that all look the same. And we can match them to neurons that have the 70% of development. And we can make a perfect match between one cluster and the other. The shape of the cluster doesn't look the same because there's a different processing. And you can do that at 50% and so on, 40, 30, and 15% development. And so just to illustrate what I want to say you is that indeed there is one blob of neurons here that corresponds to T4 and T5 neuron. But if you look at 50% development, in here you did something quite different. So those neurons, T4 and T5, they are right here. And you can see those 8 neurons now beautifully well separate from each other. They have different transcriptome because the stage when indeed they make the connection, and therefore they are going to express different transcription factor and different cell adhesion molecules that will allow them to connect to different parts of the brain. Which means that it's very nice to do the single cell sequencing in the adult, but if you want to see the full diversity of neurons that are present in the brain, you need to make sure that you're actually going to be able to do that during development at the time when diversity really gets expressed, by establishing different types of connection. Okay. So now what you are doing is try to see how we can take this neuron at this stage of development when they make connection. And we know that, for example, this T5 neuron will connect, for example, to the TM3 neurons, and try to see how those two neurons here connect to each other and what kind of additional molecules they do that. So it's a very large amount of work because, it's not simply that, neuron A connects to neuron B because neuron A expresses cell adhesion molecule X that is connecting to cell adhesion molecule Y into neuron B. It's a lot more complex than that because this combination of cell adhesion molecules that together allow two neurons to talk to each other. But now we have a large statistical power because we have those 200 neurons that exist there, and we can see how they're connecting to another, but still not trivial to be able to do that, and you are very busy trying to understand what's going on there. Let me check the time to know where we stand. Okay. And again, feel free to ask me any questions if you want to. I'll be glad to answer any question. Okay, so let me see your faces. Yes, okay. So let's see what you can see in terms of understanding what's going on. Okay. So again, we can have these neurons here, and the question will be, how can we figure out the next step in the process that I told you that we could indeed identify the neuron diversity in this brain. But the question is, how would you make those neurons different from one another? Okay, because it's nice to see that they are different from one another. They connect differently. They have different transcriptome, but how do they become so different from one another? How do they develop to become different from one another? And this is a problem which exists in every brain structure, in the fly, but also in the human. And you need to see that we have quite a deep understanding of what's going on in this species, which hopefully could be expanded to what happened in human or mouse. Okay? So I'm going to focus on one brain structure, this medulla structure that I described to you earlier. And again, you remember, I told you it's made of two regions, the region where the cell bodies are present, and the projection to the 800 columns, which correspond to the 800 pixels that the fly can detect. Okay? So the question is, how do you generate those 40,000 neurons that are going to innervate all of those neurons? Okay? So what you should know is that this doesn't happen in one day. The brain, and actually it's true in our cortex, the human cortex, doesn't develop in one day, it develops stepwise. And here, what I'm showing you is the development of not the medulla, but actually the eye itself of a fruit fly. What you have on the right side, you have a single neuroepithelium labeled in red. And what happens is that at one edge of the neuroepithelium, cells transform, they express different transcription factors, and they become photoreceptor cells. And then there is a wave of differentiations that go from right to left. And as you can see, as time progresses over a period of 2 or 3 days, you recruit more and more photoreceptor cells until you're going to consume, in a day or two from now, every part of the neuroepithelium, and everything has been transformed into photoreceptor cells. You might know that the very same thing happened in the human cortex, whereby you have radial glial cells that are formed first, and they innervate layer 6 of your brain. Then they're going to, a day or two later, transform into produce other neurons that are going to innervate layer 5, and then layer 4, layer 3, layer 2. That means you have a sequential production of different types of neurons, which are going to happen with depending on time. So now I was telling you about the eyes, and through the work of many people about 20 years ago, we know very well how the eye developed. And so it's not what I want to focus my attention on. I want to focus my attention on the development of this medulla structure. This medulla structure develops from a neuroepithelium, and the neuroepithelium is this purple region that you see here, this purple region which has a crescent shape. And this receives innervation from the eye, from the photoreceptor of the eye through the optic nerve. But what matters today is really how you're going to transform this neuroepithelium into the 100 cell types that form the structure of the medulla. Okay? And so you hear this neuroepithelium early in development, and it's going to produce all of the neurons in the medulla. As you do that, this neuroepithelium will be transformed from a neuroepithelium into neural stem cells by forming first neural stem cells at the outer edge of this crescent shape, okay, and produce neurons which are going to bud off beyond the screen, the plane of the screen. So here is an example. You can see here you have these neural stem cells that are forming at the outer edge of the crescent, okay, and then the whole neuroepithelium will be consumed as time goes because it's going to be all transformed into those pink cells. And I can just show you a diagram of what's going on here. Again, in pink or in purple, you have the neuroepithelium, and in blue, what you have the stem cells that are formed. And as time goes, you're going to transform all the neuroepithelium into stem cells until there is nothing left from the neuroepithelium. And you're going to generate a fairly large number of stem cells. And there is a funny number here, that because the number of stem cells that you define is actually about 800, which means remember, 800 is about the number of a facet in the eye, which means the number of pixels that the fly can detect. And so it seems that maybe what you have is that you have one neural stem cell, we call them neuroblasts, one neural stem cell for each one of the columns that you have that process information from one pixel. So one idea will be that in the medulla, which is composed of 40,000 neurons belonging to 100 cell types, each one of the neural stem cells will produce the 100 cell types that surround each one of the 800 columns. Now, if you know something about neural stem cells, you can see it's hard to imagine that one neural stem cell can produce 100 different types of neurons. One stem cell produces 100 different cell types. It's hard to imagine how this can be possible, although it's almost true. What I'm going to tell you is almost that is exactly what's going on there. So how can we understand the process? So what we can do now is giving you an idea about what's going on. So you have, I'm going to make a cross section through this tissue here and flip it 90 degrees. Here I get a side view. And again, you can see here, the neuroepithelium is here. And the neuroepithelium will be transformed at the outer edge of the crescent into a neural stem cell. So now you're going to have a neural stem cell, again, called neuroblast. And neural stem cells do what neural stem cells do. They divide asymmetrically to renew themselves and to produce neurons. In this case, they produce an intermediate precursor called a GMC. This intermediate precursor divides only once to give rise to two neurons. So every time a neural stem cell divides, a neuroblast divides, it gives rise to two neurons. So at this stage, what you have just one single stem cell in this plan of the neuroepithelium. But an hour or two later, the next neuroepithelial cell will become a neural stem cell and will do the same thing, divide and give rise to two neurons. But at the same time, the first one was still able to divide. And what we're going to do is something like this, which means as you consume the whole neuroepithelium to form neural stem cells, all the stem cells will divide, keep dividing, keep dividing, keep dividing. And they're going to form, because it's such a compact structure, they're going to form these long columns of two neurons which are below each one of the stem cells. And actually, just to show you that it's not a lie, that you have indeed, if you label with GFE one single neuroblast here, you can see below it, there is this long column of neurons, of two neurons, that below they come from the same mother, the same neural stem cell. And you can see you have a fairly large number of divisions because there's quite a large number of cells like below each one of the stem cells. But here again, talking about time, you can see that you have two types of neural stem cell. You have the young neural stem cells that just got transformed from the neuroepithelium into neural stem cells, and old neural stem cells that have been done 2 or 3 days ago. And those that produce neurons, long column of neurons. These are just the neurons which have just got produced, and the neuron they got produced yesterday or 2 days ago. So you have young neuron and old neuron, and you have young neuroblasts and old neuroblasts. So two time axes there that are important for generating the neural diversity. However, the question here is that how can one neural stem cell here can generate, I was telling you, something like 100 different cell types. Now it's start to think possible that, that maybe you can have maybe 100 cells below each one of those columns. Maybe it's not 100, but it's not too far, maybe 50. And so we try to understand how this can happen. And the beauty of the system is actually we knew conceptually how this could happen from the work of my friend Chris Doe, who is in Oregon, in Eugene, Oregon. And Chris Doe, many years ago, over 20 years ago, came out with a very nice system by which a single neural stem cell could produce many different cell types. And this was actually in the ventral nerve cord, which means the spinal cord of the flies. And there is some neural stem cell here, there, a neuroblast. And those neuroblasts, when they're young, they express a transcription factor called hunchback. When they divide, of course, they're going to express hunchback into their progeny, and they're going to renew themselves. But then what happened, actually, the same neuroblast that express hunchback decided it's tired of expressing hunchback, and instead is going to switch off hunchback and switch on a new transcription factor called cripple. The name doesn't matter. But of course, the next time they divide, they're going to put the blue neurons instead of the red neuron because they express cripple. Then they get tired of expressing cripple, express green PDM. Next time they divide, they're going to put the green neuron, and then get to change fate again, express castor, express a yellow neuron. Which means, you can see again, as we saw before, a long column of neurons. The oldest one express hunchback, then crippled, then PDMs, and castor. And therefore, those 4 neurons expressing different transcription factor will likely become different because they have different transcriptome. And indeed they do. But of course, these are only 4 neurons, and we need to generate 100. So this system is beautiful, but you might need to extrapolate to make it a bit larger. So when this was discovered many years ago, 20 years ago, people got all excited because it explained, as I was telling you, how the radial glia in the human brain can produce neurons from layer 6, layer 5, layer 4, layer 3, layer 2, because it could be that those radial glia change their fate from hunchback to cripple to PDM to castor. Or in the mouse or human retina, you know that neurons are also produced in a sequential manner. First, you produce the retinal ganglion cells, the RGCs, and you produce bipolar and horizontal cells, and you produce cones, and then you produce forward in a sequential manner. So people immediately jumped onto that and to figure out whether they can find the same gene expressed there. And I must say it has been pretty much of a failure in the sense that people didn't find really good genes to express in those type of neural stem cells they could identify. And they did the same thing in many different species, and nobody really came up with a system which would be similar to that system there. Okay? So we said, okay, maybe we have been more lucky. And we have an advantage because in our system, because the neuroblasts are produced sequentially, you could imagine that in one snapshot, you could see a neuroblast express hunchback and cripple and castor, sorry, PDM and castor. And therefore, you will be in a single snapshot, the neurons express hunchback and cripple and PDM and castor. But no, like everybody else, we failed. None of those genes was expressed there. However, we did find something which was exciting because we found the very same thing, except we had to change the name and we had to have more names. In the center, we had the very same concept, but now instead of having hunchback, cripple, PDM, castor, we find a young neuron, neuroblast express homothorax, then clufus, then homeobrain, then islets, then stobipert, then dacit, then tailus. So you can see that you have a larger number of gene transcription factors they can express, which presumably can produce different types of neurons. Okay? This was not by far thorough. It was just by chance we find genes or educated guess, we can find the genes that would be expressed there. We try many antibodies and we identify those genes, and we're very happy about it. Okay? So now the question is, okay, if you have a neurostem cell or neuroblast that switch fate from homothorax to clufus to homeobrain to islets to stobipert, how does it make this switch of fate? How can the same neurons, the same cell, can first express a transcription factor, then switch its fate and express a different transcription factor? And so again, here I just took 5 of those to make it simple. And I can ask the question, how come the system progressed from homothorax to islets to stobipert to dacit? So I say the best question would be to, for example, take out one of those genes, like stobipert, and see what happened to the series if you stop expressing stop paired, if you make a mutation in stop paired. So if you do that, in fact, the neurons keeps progressing. That means it keeps going to divide, but then it's stuck into the previous temporal window, is stuck into the previous eyeless temporal window and never progress toward the dikit and telos. Okay, which means it was likely. That means that actually X, the gene stop paired, activates dikit and represses eyeless. So the same transcription factor can do both, progress the pathway by moving to the next transcription factor and repress the previous one. If you don't have X, if you don't have stop paired, you get stuck into the eyeless because you cannot repress it, and you can never activate dikit and telos. It seems here the progression of the temporal window is due to transcriptional progression, whereby you go from one gene activating the next one, activating the next one, and repressing the previous one. And it seems to be working the right way, because indeed we can see that it's almost the case for all of those genes here on this temporal series. So again, this is a picture which is really one of my favorite pictures, whereby you can see exactly what I just described to you. So it's a top view of the neuroepithelium, you remember? So the neuroepithelium is in gray here, and actually it's quite advanced because the crescent shape was here, and the first neuroblasts were forming at the outer edge, and they progress here. So this is actually the youngest neuroblast that just got transformed from the neuroepithelium into neuroblast. And you remember, the youngest one expressed homothorax. Indeed, the staining here is for homothorax. And then these were a bit older neurostem cells that express eyeless. Then older one express stop paired, older one express dikit. And I don't have enough picture color on the confocal microscope, but the last one will be telos expressing cell here. So you can really see here in the plan of the neuroepithelium that indeed what you have is actually the staining for all the transcription factors that I get. And now I don't have time to tell you about that, but we have actually extended this series quite a lot further by actually using again single cell sequencing. And now we know a complete series about 12 transcription factors that's actually going to be produced there. So what I try. What's going on? Okay. What I'm going to tell you here is that indeed the techniques that we have used, both the techniques of single cell sequencing, but also the techniques of molecular genetics by using the power of fruit fly genetics, have allowed us to find the rules of how you can generate different neuroblasts and different neurons. And the rules are simple. You have a neuroblast dedicated to each one of the columns, and this neuroblast is going to produce all the neurons that are going to be surrounding each one of the columns. If you have 800 neuroblasts, each one produces all the neurons of this 800 columns that I described to you earlier. So again, I'm basically oversimplifying a bit because it's a bit some slight tilt on the system. But overall, the concept will be that indeed, if you want to generate neural diversity, the best way is actually to use this system of temporal patterning, whereby you have a cascade of transcription factor, which is going to be producing all the neurons from that. So I want just to stop here and give credit to the people who have done the work. And Nikos is actually a postdoc who is going to become a professor in France, in Paris, at the Institut Jacques Monod, and he's the one who pioneered the use of single-cell sequencing before it became the fashion, and before we had a machine to doing that, we had to do it with some homemade devices. And then two postdoc and a student in the lab, Neshet and Felix, took over this work and did a larger set of neurons using new technology, again, for single-cell sequencing. And there is this amazing machine called the 10x genomics machine, which is not terribly expensive machine, but a really huge power for doing that. And only told you the work of those people, but there is other people at a fairly large lab where you have a bunch of other people who are doing a lot of work on this system. And one day I will tell you what we do also on ants, and because we're trying to understand the brain of ants. And of course, ants are amazing animals because they have belong to different castes. They can be the queen, or they can use the workers or also warriors. And we want to understand how the brain of these different castes is actually defined. And and how basically epigenetics control can actually make sure that you have different classes. So we do that in collaboration with Daniel Randberg at the med school of NYU. And then there's the people who left the lab and to establish their own lab at different locations in the US, in Europe, and throughout the world. Okay. So I'm going to stop here and take any questions, and be happy to stay with you and to figure out, to discuss with you what you do. If you want to know, I would like to know what you do. Okay.
Thank you, Claude, for this wonderful talk. It was very fascinating. I did not know we had so many neurons existing.
Oh, you underestimate your brain, I guess.
So we have quite some questions. I see most of them are based on the transcriptomics data that you shared. So we first have a question from Swikrut. Swikrut, would you like to unmute and ask it yourself?
Yeah, hi. Am I audible?
Yeah, you are.
Good afternoon.
Great. Yeah, so first, before I get to this question, Professor, how do I pronounce your name? Is it Desplan or Desplan?
Yeah,
I'm sorry.
No, Desplan. I mean, in France, it will be more Desplan, but because I'm from the south of the country, we pronounce most of the letters. We say Desplan.
Desplan. Okay. Thank you very much, Professor Desplan. So my question was pertaining mostly to the how you came to choosing the cluster number. So this is something, I guess, in machine learning as well. They have different algorithms, and they find different ways to decide an ideal cluster number. So here I'm guessing what you were doing was minimizing the intercluster, the sequence difference, right? But that brings me to another question, which is how this is, I mean, how useful is this, and will this apply for helping us with coming out with a connectome for the fruit fly? Because we need to make sure that a connectome is kind of very representative of the entire species. So is your cluster data from multiple organisms, or is it from within a single fly?
Okay. So again, technically, the processing of information here is done by this program called SORAT from Rahul Satija. It's a very sophisticated program that works beautifully well to do this clustering. And you have different parameters you can see to increase the clustering or decrease the clustering. Again, let me show you the picture and you're going to see what I mean by that. So I'm sure you have seen many pictures of single cell sequencing throughout. So I still share my screen. I don't share my screen anymore. Sorry, let me share it again. Okay. So you have seen, I'm sure, a lot of pictures of single cell sequencing. One of the beauty here on this single cell sequencing is, unlike most of what you have seen here, the clusters are very, very well separated from each other. It's a t-SNE, they are very well separated, which means they're really very significant transcriptional difference between those clusters. And I must say that actually our colleagues, Larry Ziprowski at UCLA, has done a very similar type of work. And we combine our data together and they completely support each other. I mean, complete similarity among those two. So again, we think it was fairly easy for us to choose a parameter to be able to get the right level of clustering. But of course, you can, for example, over cluster, you can actually change the parameter and subcluster this cluster here on subcluster. And most of the case, it doesn't lead anywhere because in fact, you keep one cluster. And even if you force it, you can see that those differences are not totally clear. Although, you know, I must say that there are some examples for some of the clusters when you could subcluster them into dorsal and ventral cluster based on very, very tiny difference. But overall, I think what you have here is, I know for sure this is actually an MI4 neuron and only the MI4 neuron. There might be two subtypes of MI4 neuron, but it's still MI1. It's a bit different for the smaller cluster. The smaller cluster are many fewer cells, and sometimes it's a bit harder. And I'm pretty sure that some of the smaller clusters might correspond to multiple cell types, very tiny, very low order cell type. So we are working very hard in doing that to make sure that you don't. So I don't think we under cluster, and I don't think there's a limited number of over clustering. Okay, I don't think we over cluster. I think we might miss some of the cluster, but again, we're working very hard on that. And your next question about how can this relate to the connector? Okay, so indeed, is what we try to do. Now we have, for example, we know that these two cells MI4 and MI9 might connect to one another. And so what allows them to make connection here? So what you have to realize, you remember, this, I didn't tell you very much about it. So you can see here, this is actually a medulla. And actually you can see this neuron here, an example is a TM neuron. It projects mostly into these layers of the medulla. In fact, you can define 10 layers in the medulla here. And in the lobula, you can define 6 layers, which means not only you know where they connect, you also know the layer where they connect. And not all neurons first connect to layer 3 here. Not all neurons connect to layer 1 of the lobula here. So that means that you already can subgroup the different neurons based on their connector. In fact, this neuron only connect to layer 3 here. Other neurons connect to layer 3, layer 5, and layer 9. So again, you can subgroup those different neurons based on their layer projection. And the question would be said, how do they know where to go to the right layer first? And how do they go to connect in the right layer to the right neuron? And I must say that so far we have not managed to achieve that, because we know which transcription factor expressed in those 2 cell types. We know which cell adhesion molecule expressed in those cell types, but it's not so simple to say this cell adhesion and its cognate in the next cell over allows the connection. Because if you make a mutation in any of them, nothing happens. That means it's not a system which is very simple. You know, the notion from Sperry claims, you have 2 adhesion molecules connecting to one another. It's true, but it's not working with 1, it works with 10. You need a combination of 10 of those, which are partly redundant to be able to see them. And if you disrupt one of them, not much is going to happen. So the point now that we try to do is to not look at pairs of neurons, but to look at dozens and dozens of pairs of neurons and hoping to define the rule of the game. And I'm afraid that the rule of the game are going to be quite different from one neuron to another because of evolution. Evolution creates synapse between neurons as evolution proceeds, and it's going to take whatever is available in those neurons to allow them to connect. And so it's not going to have a general rule that was designed 100 million years ago, and those rules apply to all neurons. No, those rules have been established through time, and therefore they're going to differ from all neurons versus neurons that appeared in the connectome in the most recent, like 10 or 20 million years. Okay? So I hope I gave you an answer that satisfies you. So,
yeah, for the most part. Thank you so much, Professor. I think there are a lot more questions, which will also kind of, there are more questions coming down here, which are related to other things I would have liked to ask you. So I will.
But I mean, I can hang around for a while and you can talk to me.
Yeah, no problem. But I will, move on to the next one for the time being. Thank you so much.
Okay. Now we have a question by Karan, which I think you answered in one of your subsequent slides. The question is, can the transcriptome of neurons change temporally? If yes, the clustering by this technique might not be correct.
Okay, so it's a very good question. And indeed, the transcriptome does change quite significantly from early to late. So what we have done, we cluster each cluster independently at different time points, which means you have 200 cluster in the adult, you also 200 cluster at 70% patient, 50. And the point will be to try to match the cluster from the earlier time to the cluster in the later time. And again, we have designed some computer program to take advantage of that, and actually we can manage to do that almost perfectly. We have almost no ambiguity. It means that every neuron you find at 50% patient, you can match with a neuron in the adult, and vice versa. It means that really it works beautifully well. So, and it makes some sense because although the transcriptome does change, it doesn't change completely. So there are some genes that remain constant. And you have this notion that you have some transcription factors, for example, that are labeling the cell early on in life and that remain expressed throughout the life of the cell. This is work mostly pioneered by Oliver Hober, who claims that there are some of these transcription factors that are really marking the cells throughout their life, and then you can recognize them. So really, there is no ambiguity in terms of, again, showing you my picture here that, and sharing my screen again. So this, this is a cluster independently from that one, but then you can, what you could do identity transfer when you can match each cluster to the other cluster. And again, you could see, for example, in the case of the T4 and T5 neurons, those 8 neurons that are here match to only one cluster here. Okay? But they match well. So we know that these neurons here are the same as those, except here they were not clustered. So the transcription does change, but we can overcome this problem and make sure to identify the different cell types. Okay.
Yeah, I think that answers the question. We have another question which says that there are so many types of neurons based on transcriptome. Are they all different functionally? If so, how many different functions are there?
Okay, so again, we have another great advantage over our system in the sense that each functional neuron is must be present in each one of the columns of the system, because you want to process every part of the visual field the same way, which means that each cartridge, each column below one of the pixels must contain all the neurons that process information. And so your question is it always the same? I do believe that each one of them is going to be present the same way. That means that each column will be processed the very same way, which means you expect to find each neuron in each one of the cartridges. So neuron can be large, can be processing different columns, but still they should be present at least once in each one of them. Now, if you find two neurons that are in the same cartridge, two neurons that look the same, that are in the same cartridge, most likely they do different things, because there is no reason to having more than one neuron. Okay? So if you have two neurons, most likely they represent two cell types. For example, the T4 and T5 neurons, there are 8 of them in each cartridge. And therefore, you can see that do perform different functions because one is front to back to front, up and down motion. Okay? So indeed, the neurons are performing different functions. Now, there is some of the debate among the people who's most likely from the vertebrates. They say, is a cell type a discrete system, or is it a continuous system from one cell type to another? And I have a strong feeling about that, in the sense I do believe that a cell type is a cell type and it's very distinct from the next cell type. And it's actually, again, clearly illustrated in the single cell sequencing, in the sense that, there is no ambiguity that indeed here, it shows, sorry. The cluster, are absolutely separated from one another. So if it was a continuous cell type, you see something between that and those two neurons, okay? And you don't see that. You don't see that because they're very clearly distinct from one another, okay? So really clearly cell types defined, well-defined, and clearly separated from all the other cell types.
Thank you for that. We have a few other questions. We have a question by Jay. I see he's there in the meeting. Jay, would you like to unmute yourself and ask the question yourself?
Hey, Jay.
It seems like he can't unmute himself. I'll ask the question for him. Does the interaction of different neuron types change their transcriptome?
Yeah, so it's also another question. And I'm going to be very dogmatic here in the sense that, in vertebrates, in the human or mouse brain, that indeed neurons are influenced by the environment, okay? Who they connect, whose activities they are going to make them who they are, okay? And this is clear. I'm going to take the position that in the flies, this does not happen very much. I'm sure it happens a bit, but it's definitely not a major factor. And again, I do believe that the transcriptome directs most of the fate of those neurons, okay? And so the activity, for example, who they connect might influence whether they survive or not survive, but I do believe that they are first defined by the transcriptome, and there is little effect of activity of them. Again, we published a paper like 2 years ago, it was in Science, where we showed that there are some neurons that have to disappear by cell death because they don't find the right partner. But they are defined, and if they don't find the right partner, they die. But they don't change their fate based on the partner. Exactly was the point of the paper, that indeed fate was there before. And the question of how you make sure you have the connection is because the cell survive only if they connect the right partner. And in this case, we could actually identify the cell adhesion molecules that are involved in this process. It's a paper by Fernandez, by my student Maximilien Courgeon and myself. Okay? It was in 2018 Science. Or 19.
I will look that up sometime. We have another question from Karan. Can transcriptomics tell us anything about connectomics?
Yeah, I see it's the same question. That can we learn something from the transcriptomics? And for now, I say no. But clearly, the answer to your previous question says it should be yes, because indeed, I say everything is in a transcriptome. But so far, we have not managed to understand how different cell adhesion molecules can control the connection to two cell types. Again, there are a few exceptions. I just mentioned this science paper where you have one example where this happens, but it's kind of a peculiar example. In general, so far, we have not managed to do that. Again, most likely because there is a lot of redundancy among the systems. That means that cell adhesion molecules are not working on their own. They work as multiple, they need multiple of them. And we rely a lot on mutant phenotype in our system. And the problem is that if you make mutation in one cell adhesion molecule, usually it doesn't lead to very much, or if any, of the phenotype. And therefore, it's very hard to figure out what the role is if you cannot find a phenotype. So we know that indeed different cell types have different cell adhesion molecule. And again, let me share my screen again and show you what I mean by that. Oh, sorry. No. So I clicked the wrong button. So let me go back to my screen and yeah. So share the screen. Sorry. So you remember I told you that this cluster can be split into 8 clusters, okay? And I show you this cluster can be split into cluster, and then you can see that in the 50% development, there is actually 8 clusters. And those 8 clusters can be defined based on the transcriptome, and we know that they go to different layers of the optic lobe, okay? They do the same thing, but they do go to different layers of the optic lobe. And we have cell adhesion molecules that seem to instruct to correspond to the one that go to one part of the optic lobe or another part. Yet, if you mutate those cell adhesion molecules, not much happens. So again, we don't understand what's going on there because I think we have to pick a way to test multiple cell adhesion molecules at the same time. And so far, it's quite hard to test more than one or two genes per mutation. Okay.
Shreesh has a few questions in the chat box and he's here with us. So Shreesh, if you'd like to unmute yourself to ask them, go ahead.
Yeah, thank you so much, Professor Desplan. The first question I had is the conversion of neural stem cells to functional neurons irreversible? And if so, how might that be made possible? Like, so that the functional neurons do not go back to stem cells?
So it happened that neurons, not so much neuron, but the intermediate progenitor can actually switch back to stem cell, but then this causes tumor, and it's only upon mutation. So normally it does not happen. So your question, I guess, is can you have a feedback from the neuron telling the stem cell, hey, I need more of these neural cell types, and therefore keep doing it. And again, I don't think there is much plasticity. So of course, this could sound like a disadvantage because we know that plasticity exists in the human and mouse brain, and I don't think it exists very much in the fly brain. So it's a disadvantage in a sense we cannot do everything, but a huge advantage in a sense that now we can focus our attention on what is possible. So we miss some of the phenomena, but we can understand the specification much better because there is little of this flexibility. So I do not believe that you can go back from one neuron to neural stem cells. And actually, even in the vertebrate brain, it's very hard because all the brain tumors that you have are not actually neural, so are not neuron becoming cancerous. Actually, it's a glial cells that become cancerous, or actually that there are the stem cells that become cancerous. So you know, when you have differential the neuron, it's very, very hard to go back and to become a stem cell again. You can force it, and there are people that try to do that, but you really have to push very hard to re-enter them in a cell cycle. So my answer would be no, you cannot go from one neuron to going back to a neural stem cell.
Okay. And the second question I had is this is pretty much talked about in the talk itself, but how much does how much does the transcriptome differ within neurons in the same group or the same cluster? So, I mean, what is the level to which you can keep reducing or increasing the resolution of the cluster?
Yeah.
To a point that the neurons will be the same.
Yeah. So the question that you asked is that again, I'm going to share my screen here. Is that how much? So how much the cells in this cluster here resemble each other? So again, it depends on the technique that you're using. When we first started doing that, we could get about 1,000 genes per cell. And of course, there is more than 1,000 genes expressed per cell. Now with a new technology, with a 10x genomics, we can get a few thousand genes. It's not the full transcriptome, but it's good enough that actually you can cluster those cells. And again, my argument will be that here you can see that the clusters are very, very well defined as compared to the next cluster over. So this is very distinct from that. There is a long distance there. And again, there is no ambiguity that this neuron is MI9. Now, is your question could be, is there several subtypes of MI9? Possibly, but they are still going to be MI9 because there's only one per columns. And again, in the case of MI9, we did find as the fact that this cluster can be split into two, the dorsal cluster and the ventral cluster, based on very, very tiny difference. But overall, I do believe that these cells are all very, very similar to one another. They really are the same cell type. There can be some small variation, but they're the same cell types.
Yes, sir. So the individual differences which might be there, the very tiny differences which might be there in the same cluster.
No, I think that the tiny difference that you mentioned are purely technical difference, or mostly technical difference. It means, you know, you capture only part of the transcriptome, and therefore you don't have the full description of each one of the cell types. However, you know, for totally different projects, we are doing lineage analysis, and we can at least, the output of the system is to find the transcriptome of a single cell. And we want to know what the single cell is. And we can actually match a single cell to a very different cluster. So there is enough information in the transcriptome of one cluster or one cell to match it to the whole cluster. So again, especially with the new 10x genomics, is really very, very powerful. We sequence quite a large proportion of all the transcriptome of each one of the neurons. It's pretty amazing actually how they do that. Sorry, Shota, I interrupted you. So
yes, sir. No. Thank you so much. Great talk, sir. Thank you so much.
I think we have one more question from Anuj. Anuj, would you like to unmute yourself? Is Anuj here? Yeah, he's here.
he's here.
Yeah, am I audible?
Yeah, we can hear you.
Yeah. So thanks a lot for such a talk. And my question is that, sir, is there any significant difference between the neuron transcriptome that have been taken from this infant versus the mature one, or this is the same? I think because there is a evolution process from this infant to this maturation. Is there any significant difference between the neuron transcriptome of infant and the mature one?
So again, I could have given another talk today talking exactly about that. So I mean, actually, I can show you some picture, and you will see what I mean by that. So you can see my screen here. Yeah. So it's actually the same picture except taken much earlier in development. And what you have here, you have the neuroblasts, which are here, and then you have the different neurons. And you can see that now the neurons are not yet blocked. In blue are the neurons as I described them to you in the transcriptome later in development. But early in development, you can see there's a long trajectory coming from the neuroblast to the mature neuron. So your question is exactly that. How does the transcriptome change from neuroblast to actually forming the mature neuron? And so you can analyze the different types of genes expressed in this process here. And you can see each one of those lines are going to lead to one specific neuron. So it was okay. You can see here, each one of those lines goes to the terminal differentiation of a, for example, TM3 neuron or MI1 neuron, MI4 neuron. And you can study this trajectory to know what exactly happens during this maturation process. So, but it's a whole new story that I will tell you another day, okay? But we do have access to that, yes. And you sense the transcriptome change quite significant.
Thank you so much, sir. Thank you so much.
Okay.
Okay, with this, we come to an end of your talk, Claude. I would like to hand it over to Pranjal of Project Encephalon. Over to you.
Okay.
Hello. So, hi, I'm Pranjal Garg, and I'm current president of Project Encephalon. I would like to ask Dr. Desplan if you would like to interact with the attendees beyond the talk, if you have time and if you want to.
Absolutely, absolutely, yes.
Okay,
No
so
problem.
if any of the attendees wants to become a panelist and come up and be on his video and talk, so you can just raise your hand and I will promote you to a panelist. So please go ahead. In the meantime, I can, okay, so I have one attendee over here, Mr. Kama. Shreesht is here. Okay, Gagandeep is here. She wants to talk to. In the meantime, like everyone would want to ask what to do to come as an intern in your lab.
Sorry, can you use the question again?
What to do to come as an internship or an intern student in your lab? And, I mean, that's the question everybody would want to ask. Yeah.
You know, nowadays it's not so easy because of the pandemic, and so hopefully things will get better. And especially now coming from India is almost impossible. I think the administration banned travel from India, so it's going to be tough until you guys solve your plan. Although, you know, I must tell you that, you know, there is a big problem in India right now, but you are where we were 6 months ago. You know, we have a much smaller country, and still we managed to kill more people in our country than you do now these days. So hopefully it will not go further in India, but you know, we at some point there was 5,000 or 6,000 people dying every day in the US, and you know, they have a very good health system, yet they were completely overcome. So it's bad what happened in India right now, but you know, we don't have any lesson to give you. So so far the travel ban exists and it's going to hold for a while, I'm sure. Hopefully we'll be able to provide you with a bunch of vaccine, which, by the way, many of them are produced in India themselves. So you should be able to keep them and vaccinate a bunch of people. You know, we now have vaccinated about 50% of or 60% of the American, and it's making a huge difference. I mean, basically everything, all the cases are dropping. People don't die anymore. I think 2 or 3 states yesterday reported there was no deaths from COVID in their state. So it's amazing. So vaccination works very well. Same thing in England, same thing in Israel. So hopefully, I mean, it's a bit more difficult in India because you have 1.4 billion, 1.4.
Not 1.4, I guess 1.1 billion, around 1.1 billion people we have. So
Okay, yeah,
that's good.
So anyway, it's a huge number to do, and but you know, hopefully you will get there. So to go back to your questions, you know, it's difficult for you guys because there are so many of you scientists, and again, there are great scientists in India. You know, the country produces many very, very good scientists. It's a bit hard to come out, you know, to say why am I good and why should I go to your lab to be able to perform an internship. So one way, the official way, is to get into a PhD programs. And you know, many of the PhD programs in the US do accept international students. For example, we have actually quite a large number of students from different countries, including every year we have about, since this year we have 14 students. I think 2 of them are from India, and they come from India. The question is, how do come out of the individual? I mean, how do you become individual? And again, you have to make sure that you try to explain why you are so special, and and hopefully come from a good lab in India if you can, and get some recommendation. And it's good also to have contacts with some people in the US or in France or in England or in Germany who you can have contact with. So often contacting people one by one, it's important. And I don't mean, sending 10,000 email or very generic email, say, I love your work, I want to work with you. No, it doesn't work. You can, for example, target one single professor and say, I read your paper and there's this question. Can you tell me why, why this happens that way? The kind of question you ask me in a talk, but ask a scientist in an email. And usually people will actually be responding. Not all will respond, but many of them will respond. And you can engage in a conversation. And then it greatly helped, to say, this woman sounds very, very smart, and then we should try to see what we can do to help this person. So I think you have to come out of the middle ground and try to stand out of this. So contacting people individually and basically customize. Whenever an email, never send generic email. Always send very, very, very defined email. So I hate when I get email, dear doctor, and I have a different font and bold Desplan. I love your work on a different font, neural diversity in flight. So I hate it. I don't respond to it. But if you send me an email that looks like you indeed work on that and figure out what we did and whether you're interested, I think this can make a big difference. But then again, applying to a PhD program is very good. And also what you should know is that once a good PhD student comes from one place, we tend to attract other PhD students from the same place. So if you do well, and your friends 2 years later might have a much higher chance of being recruited. So, and again, when you come to a PhD program, you get fully supported. So you get a fairly good salary that allows you to survive, even in very expensive New York. So so money is not an issue. Visa can be an issue, but I think with the new administration, I think it should be quite a lot easier than before. So.
Fish, do you want to go next and ask something?
Yeah, I think the most burning question which we had, Pranjal put it across as to, but the question which I had is like, in this scenario where a lot of experimental work is not really possible because of COVID, how exactly do we make a solid case for our PhD applications once we try to apply abroad? If we
you are in a difficult position because of COVID.
Yeah.
Yes, I mean, it's a whole generation of scientists is suffering. Hopefully, will not last too long. In the US now we are back to normal. I mean, I think we're still low occupancy but overall, it's not an issue. People don't stay that long. Instead of working on their computer, they go home to work on their computer. But I don't think. I mean, since May now, so it's almost a year from now, May 2020. We are back to normal and we can work hard and do that. In your case, it might be a bit more difficult, and because people have to be a lot more careful. And so, take your time and delay your application by a year. And, you can do things which are not in the lab. You can study.
Mm-hmm.
There is a rule. You spend 1 day in the library, you can save a year at the bench. So, reading, knowing what's going on is very important. You can learn bioinformatics. And again, nowadays, if you want to be a PhD student, bioinformatics is absolutely essential. You cannot escape it. You need to be a bioinformatician. I'm not, and I do suffer from that. I understand it, but I wouldn't be able to do it. But my people are very, very good, and they can be. You don't need to program, but you need to understand and to follow the process. So learn how to be a bioinformatician. Read. It's very important. Okay, and try to see what is of interest to you. So don't waste your time, and I think it's going to pay off at the end. Because we know that you're not often in a very easy situation in terms of even in a normal situation. The lab in India are not very well funded for most part, unless maybe you are in Bangalore or stuff like that. But many labs are not very well funded, and therefore we know that it's difficult. But if you show that you have an intellectual content, I think this can make a big difference. And again, what you're doing is perfect. Invite people to come and try to make contact with them, because now you can it makes life much more personal, so it's easier. It was a very smart move from you, so what you have done.
Gagan, would you like to ask something?
Yeah, am I audible?
Yes, you are.
Yeah.
Okay, so diverting from the academic questions, like I have heard about your, like you like to chat about your trips to India. Like I want to know, I want to know like something like about like, have you ever visited India and the places you like or something, you know?
So I did visit India. Actually, it was my first trip outside of Europe. It was in 1978, and I went everywhere. We went to the Rajasthan. We went to the south. We went to Calcutta, and we went to the Ganges River. So we traveled quite extensively. It was very hard, because India is not an easy country. It's often very poor people. It's a lot of energy, a lot of people, and but it was fascinating for me. And then we went to the south, which is a bit easier to, seems less crowded, but then the food was impossible to eat because it was so hot. But we really enjoyed it. And then I came back a number of times, mostly for work. And so I went a few times to Hyderabad. I went a few times to Bangalore, and so I always try to take advantage of that to visit the neighborhood. And again, I'm impressed with what people can do with sometimes not the greatest amount of funding, but people still manage to do well and to produce amazing scientists. So, so money is not the whole issue. I think mostly it's a question of organization and again, to make contact with the outside, because it's very important to know who exists and what's going on there, and not to simply wait for the paper to come out. And you need to know what's going on, what's significant. So I do enjoy traveling to India whenever I can, and again, fascinating country. Then I went to Sri Lanka a few years ago, and it's kind of a smaller version of India, which is amazing because it's the same thing, but much less crowded, much less crowded. So it's much easier to travel also. So, because I remember one day taking the rickshaw to go to the airport in Hyderabad, and God, it was the most scary experience in my life. Trafficking between bus and trucks and bikes and oh my God. And I went to Calcutta many in 1978, and this was most amazing because such dynamic city, so much full of energy. Again, a lot of poor people, but so much full of energy. So I enjoyed it also. I want to go back. I've never been back to Calcutta.
Svikrut, would you like to go next?
Yeah, you can you hear me, and you can probably see me as well, right? Yeah.
Yeah.
Cool. Looking a bit like a convict with my pandemic haircut and my choice of shirt. But if you don't count that, then
oh yes.
Yeah.
Yeah. So short hair and orange like today. So are you in jail or not? No.
Am I in jail? No. Yeah, I'm technically in jail because we have a lockdown going on and I'm stuck at home. So I guess you can call that locked up inside house, right? So yeah, my question again, sorry, I guess, Gagan's question was very interesting, but I'm going back to a little bit of an academic question here because like many of us, we're very concerned with our careers and other things during this pandemic time. And I recently became a PhD student. I'm currently a PhD student in IISc Bangalore and working with Dr. Supratim Ray. So my question to you, Professor Desplan, so the thing is, even within neuroscience, it's a very vast field. I really like it, but I'm not able to identify exactly what I want to dedicate the rest of my life towards. And maybe a lot of neuroscientists for that matter, they have different areas that they cover. So my question to you is more of the sort, once I complete my PhD or something like that, how easy or difficult is it to migrate between one area to another area within neuroscience? Would a postdoc opportunity open up for me if I am considered to be a little more away from the research area of the guide? Is it possible to, yeah, that's my question.
So I have a very strong feeling about that. And I do believe that a postdoc should be very different from the PhD. To me, PhD is a training period, which means where you're going to, and again, it doesn't matter what you do, as long as you work with a very good PI and you get good training, thinking how to be very rigorous, to be very, to read, to discuss, to interact. This is what's important. You can work on bacteria, it's fine. You can work in yeast, it's fine. You know, one of the best person who went through my lab, she was actually a yeast cell biologist. She came to my lab and she did some of the most amazing electrophysiology. We don't do electrophysiology. She's the only person who did it. So anybody can do anything. This person is smart. Okay. Now she's a professor at Columbia. So
wow.
I think you should not worry too much about what you do as long as you do good work during your PhD. Okay. So I mean, it's better if you do basic work, you know, trying to find whether you know, this plant can cure cancer. I mean, try to do something, you know, more basic, and then you can apply it to anything you want as long as it's very good. So you know, many fellowships that are, you know, the most prestigious fellowships, they require that you change field. Okay, that means you cannot go from, you know, neurobiology to neurobiology. You at least you have to change organisms or to change what you proceed. So we want you to gain more experience. And so one is training to try to learn how to be a scientist. The next one is to basically what you're going to do for part of your life, okay, as a postdoc. So for your postdoc, you choose what you're of interest. But by then, you know, you spend 4 or 5 years doing your PhD, you read a lot of paper, you heard a lot of talks, you know what will be of interest to you. For now, choose the best person in your department and work with this and try to have good colleagues, talk to people, travel whenever you can, interact with people, do exactly what you're doing right now, okay?
Thanks a lot. That's very reassuring to hear from you, Professor Desplan. So I will focus on trying to cover as many areas as possible, get a good breadth for the field. And like you said, yeah, even I have certain interests and I will see, most likely it will be kind of different from what I'm doing in the PhD project. So I think I'm on the right track then. Thanks.
And again, it's not so hard to find postdoc. Again, if you have done some decent work during your PhD, it's not so impossible to find a postdoc in Europe or in the US or in England. Again, you need to publish some paper in decent journals and then be convincing that you can explain what you are doing. So maybe not in the top labs, but I think if you present yourself in a good way, you know, most of the, for example, in my lab, I try to choose people who can, because I have a fairly large lab and I cannot support it only for money from NIH. I need people to come with a fellowship, at least for a year or 2 or 3. So I try to tend to select people who will have a fellowship. And it's very hard for an Indian citizen to get a fellowship in the US. There are some possibilities, but it's very hard. So this is a disadvantage, but if I find somebody who looks very bright, I mean, I will still kind of, I mean, try to look into that. So.
Yeah, I think a lot of us have, even at the level of PhD and things like that, right? Struggling, especially with the pandemic, it has made things a little tricky when it comes to applying and going abroad.
Yeah, but I think, you know, pandemic is not going to be forever, hopefully. So hopefully within a year, we should be all out of it. So again, I hope in the,